It's kind of weird. My internet is kind of slow. Okay, stars. So for part two, we will focus on differential expression and differential methylation of genes using microarray data. So this is an overview of the microarray mechanism. So microarray is kind of an old way of measuring gene expression before the rapid development and adoption of RNA sequencing. So the basic idea is shown here. Suppose you have cell cultures from different conditions. Say you have normal cell cultures versus cancer cell cultures. And you obtain their mRNA and cDNA. And you label the cDNA from different conditions with different fluorescent probes. So in this case, for example, we label cancer cDNA with red probes and normal cDNA with green probes. And the idea to measure gene expression is through hybridization on a microarray or a chip, where each slot here, each probe contains a gene sequence. So if your CDNA, for example, cancer cells expresses a certain gene, and the corresponding cDNA will hybridize with that gene probe on the chip, and it will show you a color. In this case, it's red. So when you see a slot is red, that means that gene is specific to cancer cells. And similar idea, similarly, that like if a slot is green, and you will know that the gene is specific to normal cells. And if it's yellow, which is just a combination between blue and red, and you know that this gene is present in both cancer cells and normal cells. So on the right part, it's the data processing workflow for microarray data. So from your hybridized gene chip, you do a scan and you get a DAT file that contains image information. And you process the image to get a CEL file, which contains the intensity value of each probe. So the color intensity value for each probe on the chip. But you will also need a CDF library file that tells you which probe belongs to which gene. And then you can get a conventional expression matrix, like with patient in the columns and genes in the rows for downstream analysis. So for part two, question one, we will provide you with an already processed microarray data that contains 60 tumor samples and 10 normal samples. The expression matrix is already log2 transform, and if you read it in, it will look like this. So patients are in columns, and tumor samples starts with TCGA, and normal samples, I think, start with just the string normal. And genes are in the rows, and each cell contains the log2 expression value. So question one asks you to do a k-means clustering using separating this 70 samples into 3 clusters to determine whether different glioblastoma subsites exist. So if you doing this clustering, and if you observe that the 10 normal samples always group to one cluster, and and among the 60 tumor samples, some of them always group to one cluster, and the others group to another cluster, and this is indication that different glioblastoma subtypes exist, and you will be asked to use all genes and top 200 variable genes to perform k-means clustering. So I won't talk about how exactly we should do k-means clustering because you already did that in homework 2. Just be careful that for all genes, you use all the rows or genes in the rows. And for top 200 variable, top 2000 variable genes, you need to calculate the variance of each gene yourself, rank them, and choose the top 2000 variable genes to perform k-means clustering. And for this kind of large data set, I would recommend you use the following parameters. And star equal to 10 and the maximum iteration number being 100. And starting from part 2, question 2, you will be asked to perform differential expression using LIMMA. So LIMMA is just like the ESIG tool, a bioinformatic tool that allows you to figure out gene differential expression across different conditions. And for LIMMA, the idea for LIMMA is really simple. LIMMA just fits a linear model for each gene. So it's linear regression showing in the formula here. So Yij here represents the expression value for gene J in sample I, well, beta 0 is the baseline expression for gene J, and Xi is the covariate for sample I. So for example, if you want to know if the genes that differentially express comparing tumor samples to normal samples, Xi should be an indicator variable indicating whether this sample is tumor or normal, so 1 or 0. And the beta J is the difference of expression in this gene comparing tumor to the normal samples. And as you can see, beta J will be the magnitude of expression change comparing two conditions, while the significance of this coefficient will be the significance for differential gene expression. And you use p-values of beta J to obtain to call differential expressed genes. And of course, since we are fitting this model for a bunch of genes, maybe tens of thousands of genes, and multiple testing correction will be needed. Okay, I will answer your question at the end of this section. So specifically for this question, we are asking about what are the differentially expressed genes comparing the two tumor subtypes. So you need to first create the variables to indicate whether a sample belongs to subtype 2 or subtype 1. So say we use subtype 1 as the reference group, and the gene expression is the mean log2 expression will be for a gene is beta 0. And in the reference subtype. And in subtype 2, which is sort of like the test subtype, you will have its mean log2 expression as beta 0 plus beta J. It's really just linear regression. And to fit LIMMA, you need to first use this model.matrix function to create a design matrix. So this is an example of a design matrix. You should have, because you have 60 samples, you should have 60 rows here, and 2 columns. One column is the intercept, which is 1, always 1 for each sample, and another column corresponding to the covariate value xi, or specifically in this case subtype 2, indicating whether the sample belongs to subtype 2, like 0 for no and 1 for yes. And you use this model matrix, just like linear regression, to feed the LIMMA model. And similarly, like DSIG2, you will get a result table. And you use the cutoff shown here, FDR smaller than 0.05 and absolute log2 change over 1.5 to call differentially expressed genes. And for question 3, we are asking you to do pretty much the same thing, but it's just differential methylation. So we will provide you with similar to an expression matrix, this time a methylation data set. You should basically do the same thing as question 2, but a few things to know here is that not all samples have methylation data, so you have fewer samples for question 3. So you can't just use the same model matrix with 60 rows. You need to create a new model matrix, and remember to extract the correct subtype information from your Q1 clustering to build a model matrix for question 3. And another thing is that the methylation data is a fraction from 0 to 1, but limma takes values from negative infinity to infinity. So you need to first logically transform the methylation data so it ranges from minus infinity to infinity. And you can really just search online how to do a logic transformation. And then you just use limma as Q2, question 2, to get differentially methylated genes and with the same cutoff. And then the question asks you how many genes are both differentially methylated and differentially expressed. And you just use this function intersect to get the result. And I know this question is kind of tricky. It also asks you like that, is DNA methylation associated with higher or lower expression of this gene, differential genes? So you can really just choose your own approach to answer this question. There is no and absolutely fixed answer. But one possible approach you could consider is like, for each differential gene you get from running this intersect command, you calculate the association between its methylation and its expression. And you will get a correlation value for each of the gene. And you may check is the majority of the association positive or negative. Another possible approach is, for these genes, you just plot their methylation log-fold change against their expression log-fold change and calculate the association and check if it is positive or negative. And finally, still question 3, it also asks you, how many differentially expressed genes have epigenetic cause? It's kind of vague, I know. And what you should really be doing is check how many among the differential genes you get from the intersect command have opposite differential and the methylation direction. One simple way is you take the product of their methylation log-fold change and their expression log-fold change and see if you get negative value or positive value, and you count the number of negative